So let me ask you something. Have you ever really thought about what clean means? I mean, we throw that word around all the time, right? But the science behind it, well, it's this incredible, invisible battle that's happening all around us, constantly. Today, we're going to pull back the curtain and look at how we actually control all those tiny organisms. Okay, just for a second, picture your car. You probably think it's pretty clean, right? Well, get this. A recent study found that this center console, yeah, right where your coffee cup sits, can be home to over 500 microbial colonies in a tiny little spot. That's way more than the steering wheel, even more than the door handle. Kind of gross when you think about it. So yeah, it's pretty clear we're surrounded by this invisible world, and we're in a constant fight. But the good news is we've gotten really, really clever at managing it all. So the big question is, how do we fight back? How do we pick the right weapon for the right job? And that's because clean isn't just one thing. It's not an on or off switch. It's more like a whole spectrum of options. Think about it. What you'd consider clean for your kitchen counter is definitely not what a surgeon considers clean for their tools. It all depends on what you're trying to do. Okay, let's break down these terms because they're not all the same thing. At the absolute top of the pyramid, you've got sterilization. That's the total wipeout. We're talking complete elimination of every single microbe from an inanimate object, a fomite, like a surgical tool. A step down from that is disinfection. This is like using bleach on your countertop. It kills most of the microbes, but not necessarily everything. Then there's sanitization, which is what a commercial dishwasher does. It just gets the microbe count down to a safe level. And for our own bodies, our living tissue, we use antisepsis for cleaning a cut or degerming, which is literally just physically removing germs, like when you wash your hands. And this is a perfect example of why those different levels matter so much. In a hospital, right? A scalpel that's going to go inside a patient's body is a critical item. It has to be completely 100% sterile, no exceptions. But a stethoscope that just touches your skin, it's a non-critical item. It needs to be clean for sure, but sterile? Not necessary. See, it's all about the level of risk. All right, so how do we actually achieve all these different levels of clean? Well, we have a whole arsenal of germ-fighting weapons we can use. Let's take a tour of some of the big guns. And pretty much everything we use falls into one of two big categories. We can either use physical forces or we can use chemical agents. Let's dive into the physical methods first. Okay, when you need something to be absolutely positively sterile, this is what you use, the autoclave. It's the gold standard, the heavy artillery of clean. What it does is use super pressurized steam to crank the heat way past boiling, all the way up to 121 degrees Celsius. At that insane temperature and pressure, absolutely nothing can survive. Not bacteria, not viruses, not even those ridiculously tough little things called endospores. Obviously, you can't just throw your food into an autoclave. So that's where a gentler approach to heat comes in, pasteurization. You've definitely heard of this from milk. You've got two main kinds. There's high temperature short time, or HTST. That's the stuff in the refrigerated aisle. It kills the bad germs, but keeps the taste pretty much the same. Then you have ultra high temperature, or UHT. That's the milk you see sitting on a regular shelf. It's stable for months, but that super high heat does change the flavor a little bit. So it's a classic trade-off, taste versus convenience. Now, heat isn't our only physical weapon. We can also use radiation. UV light is a great example. It can't go through stuff, so it's mainly for surfaces. But when it hits a microbe, it goes right for the DNA. It basically zaps the genetic code, forcing these two little bits called biamine bases to stick together when they shouldn't. This creates what's called a thymine dimer, and it completely scrambles their genetic instructions, leading to deadly mutations. It's like breaking the zipper on their genetic code. All right, let's switch gears and talk about our chemical arsenal. Physical methods are great, but sometimes you just need a chemical. And most of these chemicals work in one of two main ways. They either mess up the microbe's proteins, causing them to unravel and stop working, that's called denaturing, or they just straight up attack the outer wall of the cell, the membrane, until it falls apart. And I guarantee you know a bunch of these already. We're talking about things like phenolics, that's the stuff in Lysol, halogens like chlorine bleach or iodine, alcohols, which are the backbone of every hand sanitizer out there, and even simple soaps and detergents, which are technically called surfactants. They play a huge part too. Okay, so we've covered everyday cleaning, standard stuff. But what about when you're dealing with the most dangerous pathogens on Earth? What happens then? This is where the science of microbial control becomes literally a matter of life and death. In the world of biosecurity, there's one number that means you are at the absolute highest level of risk. That number is 4. That's for Biological Safety Level 4, or BSL-4. This pyramid shows you how it all works. Down at the bottom, BSL-1 is for low-risk stuff, like harmless strains of E. coli. As you climb the pyramid, the danger and the safety measures get way more intense. BSL-3 is for serious diseases like tuberculosis. And then, all the way at the top, you have BSL-4. That is reserved for the absolute worst of the worst. We're talking about things like Ebola. And just listen to how the CDC describes what's in a BSL-4 lab. These agents are, quote, the most dangerous and often fatal, easily transmitted by inhalation, and cause infections for which there are no treatments or vaccinations. I mean, that is absolutely chilling. To work with these things, you need maximum containment, like those full-body pressurized spacesuits you see in movies. Okay, so we've gone from a BSL-4 lab, the most secure place on Earth, all the way back down to something much more personal. The power that is literally in your own two hands. Believe it or not, one of the most effective tools we have is just proper hand washing. Remember that term degerming? This is it in action. You're not trying to sterilize your hands, you're just physically washing the microbes off. And the CDC makes it super simple with 5 steps. Wet, lather, scrub for a good 20 seconds, sing happy birthday twice, then rinse and dry. That's it. So the next time you're standing at a sink, just think about the incredible science behind that simple action. You're not just washing your hands. You're performing a targeted act of microbial control. You're protecting yourself and everyone around you. So it makes you wonder, what invisible world are you controlling?